Now we are going to deal with the organelle which is common to both prokaryotic and eukaryotic cells, that is ribosomes. Alright. Now we have studied till now that in prokaryotic cells there are few organelles which are absent because it does not have membrane bound organelles. Ribosome is not a membrane bound organelle, please remember that. Ribosome is the site of protein synthesis. It was first discovered by George Palade. Now because I have said that it is present in both the prokaryotic and eukaryotic cells, so we need to see that in a prokaryotic cell and in a eukaryotic cell how this ribosome is present. Though it is present in both, but it is not similar in both. In prokaryotic cells you would find a 70S ribosome, and in a eukaryotic cell you would find an 80S ribosome, yes. Now what is the difference between 70S and 80S ribosome is that this S stands for Swedberg's constant, okay. Remember the name, it is Swedberg's coefficient or constant. Swedberg's coefficient represents the sedimentation, basically, how these units of the ribosome, or the ribosome for now because you are not aware of units, the ribosome for instance, how much is the sedimentation rate, that is the sedimentation coefficient of the ribosome. This is 70 Swedberg units and this is 80 Swedberg units, which points that there is a difference between sedimentation coefficients of the both, and because of that we are going to find a difference in the ribosomes. Now because both of them have to form a similar function, so the structure basically would be similar, the only difference being that the composition of the ribosome would be different, because of which the sedimentation would be different, and that composition, for that we need to understand that what is a ribosome made up of. A ribosome is made up of basically ribonucleotides, okay. In case of eukaryotic cells, it is synthesized in nucleolus part of nucleus, along with few ribosomal enzymes and rRNA, that is ribosomal RNA, and few ribonucleotides. It is going to be present in the form of non-membrane bound organelle, which would be acting as a site of protein synthesis, which is responsible for formation of proteins from the messenger RNA. Now if you are aware of the basics of genetics, you must be knowing that it is the messenger RNA from the DNA transcript that is being formed in the form of messenger RNA. The information is carried to the proteins, and that information has to be carried in the form of proteins in such a way so that the messenger RNA, whatever information is present on messenger RNA, that is properly translated into the polypeptide chain. Now these are heavy terms. All you need to understand is that what is a ribosome basically? It would be the site where the protein synthesis would take place. That means the polypeptide chain would be formed from the information which is written on mRNA. Now let us see the structure of ribosome. First of all, a ribosome is formed of a large subunit and a small subunit. The large and small subunit, they come close to each other when it is about the process of translation, which is formation of proteins from the messenger RNA. As you can see, the two subunits, they are separate, but whenever they have messenger RNA in between, that is known as a ribosome. When the both subunits, they come together, they come close to each other. Now in case of prokaryotic cells, we said that the ribosome is 70S and the large subunit is 50S. That means if we see the sedimentation coefficient of this large subunit, it would be 50S, and the smaller subunit would be 30S. Don't get confused that 50 and 30 add to 80, how come this is 70? Individually the sedimentation of each subunit is taken into consideration, and that happens to be 50 and 30, and it has nothing to do with 70. As in the case of eukaryotic cells, you would see that the ribosome is 80S, where the large subunit would be 60S and the smaller subunit would be 40S. Again, as your mathematics knowledge must be telling you that this does not add up to 80. Same thing as it applies on the prokaryotic ribosomes, it applies on the eukaryotic as well. And you get to know that what is the basic structure of ribosome. Next, what we are going to see is that what are the functions of ribosomes basically? Before that, we are going to see structure again, the bullet points that we have over here, that it is made up of proteins and RNA. As I told you, it is made up of ribonucleotides and a few proteins which are associated for the formation of ribosomes. There is no membrane present, that is the second point. Most numerous organelle. Now you have to remember that in terms of location, the ribosomes are most numerous. That is, you would find many ribosomes in a cell. Firstly, they would be present on rough endoplasmic reticulum, that would be the actual site of protein synthesis. Along with that, they would be present as few small particles where the proteins would be required inside the cell, alright. The intracellular usage proteins, they would be synthesized by freely scattered ribosomes. And apart from that, in a eukaryotic cell, mitochondria, and if it is a plant cell, chloroplasts, they also have ribosomes for their own protein synthesis, and those ribosomes, please remember, they are 70S ribosomes. So the rest of the ribosomes, they would be 80S if we are considering a eukaryotic cell. As you can see, this is the endoplasmic reticulum with small rough surface. That appearance of rough surface is due to the presence of ribosomes. Now the last point is that it is made in nucleus, in the nucleolus. What about the ribosomes which would be present in prokaryotic cell? It is believed that the mesosome is responsible for ribosome synthesis in the prokaryotic cells. One thing you have to remember. Next thing is that what is the function of ribosomes? As said earlier quite briefly, that protein synthesis would be the main process which would be carried out by ribosomes. It aids in protein synthesis, first of all. Secondly, free ribosomes make proteins used by the cell. As I told you, the intracellular proteins which would be used up by the cell, they would be produced by the free ribosomes that would be scattered inside the cytosol of the cell. And lastly, ribosomes on the rough endoplasmic reticulum, they make proteins for the export to other cells, alright. We did study in Golgi apparatus that the proteins which are formed in rough endoplasmic reticulum, they are properly packaged and then they are transferred out in the form of secretory vesicle. It goes and gives out the proteins which are formed inside the cell, so that those proteins which are formed, they can be used outside the cell. And as we have discussed, the cell type in which they are present, prokaryotic and eukaryotic cells, plant and animal cells. You would find ribosomes in all these places, and these are the points that in any case you forget anything, you have to remember these bullet points in terms of ribosomes understanding. Now the next thing that we deal with is that because ribosome, time and again we are assigning one function only, that the function of protein synthesis is, ribosomes have specific sites which act as crucial component of the protein synthesis process. Apparatus of the protein synthesis has ribosomes in it, and those ribosomes, they have specific sites over where different types of RNAs would come and attach themselves so that a polypeptide is being formed. You need not to get into the details of the aspect, but all that you have to remember is that what are the sites. So first of all, it has an E site, it has a P site, and it has A site. This is a prokaryotic ribosome that they have shown. This is the mRNA binding site that is in the smaller subunit. As you can see, 30S subunit is the smaller subunit. Messenger RNA comes and binds in the smaller subunit, that at the mRNA binding site. Apart from that, the first tRNA that comes and joins at the A site, that is amino acyl site. You know, transfer RNA undergoes amino acylation or charging. After charging, it comes to the larger subunits A site and joins over there. Then what happens? It is shifted to the peptidyl site because next tRNA is ready to join over here. Then it moves to the E site from where it is gone, and the ribosome moves further according to the mRNA binding site. If in all you can't remember the things because you cannot associate, if you haven't read the protein synthesis till now, what you have to remember is that what are the sites which are present in the ribosomes. For now, at least for now, when you are aware of what is transcription and translation, the genetic aspects, then you will get to know that what is the purpose of these sites, how the ribosomes and the tRNA come in association with each other, and how a polypeptide is being formed. So we have 4 sites that we have to remember, and for consideration purpose we have taken in consideration that a prokaryotic ribosome. Similar things happen for a eukaryotic ribosome as well. And in the end, we are going to take into consideration a eukaryotic ribosome, that is mammalian ribosome we have taken for consideration. It is an 80S ribosome. 4.2 into 10 की power 6 Daltons is the actual size of this ribosome. Now this mammalian ribosome, as you must be knowing, it is also having two subunits. Now it is having nucleotides inside it, as I told you in the beginning. The two units, first larger one would be 60S and the smaller one would be 40S. Now for remembering purpose, you have to remember that there are 49 proteins in the larger subunit and there are 33 proteins in the smaller subunit. Apart from that, how much ribosomal RNA is present inside each subunit, that is for remembering. It would be having 28S ribosomal RNA along with 5.8S ribosomal RNA along with 5S ribosomal RNA. Each S represents again that Swedberg coefficient, and apart from that we have 18S ribosomal RNA, and there is mentioned the number of each ribosomal RNA, how many nucleotides it is having. So we get an understanding that what is the difference between a prokaryotic and eukaryotic ribosome, what is the function of ribosome, what is the structure basically that enables the ribosome to be the site of protein synthesis. Ribosome is a very important organelle, and one thing you have to remember that while studying ribosome, there are two terms which are to be remembered. One is polysome and the second one is chaperones. And with understanding of these two terms, our understanding of ribosome would be complete. First of all, what is a polysome? On a messenger RNA, you know messenger RNA is a long ribbon-like structure. On a messenger RNA, when many ribosomes assemble close to each other for the translation purpose, that particular condition is known as polysome. Supposedly, over here I am going to represent it. This is messenger RNA. And ribosome 1 comes over here, second comes over here, third comes over here. So this particular arrangement of ribosomes, where many ribosomes come close to each other and form a structure where the mRNA is having many ribosomes, that is known as a polysome, alright, or the polyribosome. Second term that I told you that you need to understand is chaperones. What are chaperones? Chaperones are those proteins which enable the formed proteins or the polypeptide to move out of the cell and get transported to mitochondria. So there are proteins along with ribosome which play an important role, and they are known as chaperones, which are responsible for formation of polypeptides so that they can be formed properly, bringing close the two subunits, and formation, after the formation of protein is completed, those proteins can be transferred out of the cell. So this is all about ribosome, its understanding. I hope you are quite clear with what ribosome is and what is its function, because this is going to be a great deal of knowledge in your understanding of genetic mechanisms of translation and transcription.